Hello viewers, today we are going to see about nucleic acids. Nucleic acids are macromolecules. The nucleic acids include both DNA, that is deoxyribonucleic acid, and RNA ribonucleic acid. They are found in all cells and viruses. They are the key molecule for carrying genetic information. Composition of nucleic acid. Nucleic acids, DNA and RNA, consist of phosphoric acid, pentose sugar, and nitrogenous bases. Phosphoric acid, the formula for phosphoric acid is H3PO4. That means it contains 3 hydrogen atoms, 1 phosphorus which is located at the center of the structure, and 4 oxygen atom, out of which 1 oxygen, it is a divalent oxygen atom. One is the pentose sugar. Pentose sugars contain 5 carbon atoms. Examples for pentose sugar includes ribose and deoxyribose sugars, whereas this ribose sugar is present in RNA and deoxyribose sugar is present in DNA. Nitrogenous bases found in nucleic acid are aromatic heterocyclic compounds. The nitrogenous bases are of two types, purines and pyrimidine bases. Purine bases examples include adenine and guanine. Both adenine and guanine are present in DNA and RNA. Pyrimidine bases. Pyrimidine bases include cytosine, thymine, and uracil. Cytosine, it is present in both DNA and RNA. Thymine, present in DNA, and uracil, it is present in RNA. Let's see the difference between DNA and RNA. DNA, most of the DNA is found in the chromosomes of the cell, but some DNA is also found in the cytoplasm. Most of the RNA is present in the cytoplasm. A small quantity is also found in the nucleus. DNA contains deoxyribose sugar, whereas RNA contains ribose sugar. The nitrogenous bases found in DNA are adenine, guanine, cytosine, and thymine, whereas the nitrogenous bases found in the RNA are adenine, guanine, cytosine, and uracil. The main difference between DNA and RNA in nitrogenous bases is that instead of thymine, RNA will contain uracil and other bases like adenine, guanine, and cytosine. It is present in both DNA and RNA. In DNA, adenine pairs with thymine and guanine pairs with cytosine. In RNA, adenine pairs with uracil and guanine pairs with cytosine. In DNA, purine and pyrimidine content are equal, whereas in RNA, purine and pyrimidine content are not equal. DNA is composed of large number of nucleotides, which may be up to 4.3 million base pairs. RNA is composed of less nucleotide, which may be up to 1200 base pairs. In DNA, pairing of bases is found throughout the length of the molecule. In RNA, pairing of bases is found only in the helical region. DNA is a genetic material in all organisms, whereas in RNA, it is a genetic material in viruses. Example is retroviruses. DNA on replication forms DNA and on transcription forms RNA. RNA do not replicate or transcribe. DNA undergo mutation, whereas RNA do not undergo mutation. DNA is stable to alkali, RNA is not stable to alkali. DNA gives green color with pyronine, whereas RNA gives red color with pyronine. Let's see the properties of DNA. The properties of DNA includes denaturation and renaturation. Denaturation, it is a process in which two strands of DNA are separated. During denaturation, the hydrogen bonds formed between the base pairs are cleaved. Denaturation is brought by agent called denaturation agent. Denaturation agent may be physical agent like heat and chemical agent like acid or alkali. Let's see the effect of denaturation. The first effect is hyperchromic effect. Nucleic acid absorbs UV light at wavelength of 260 nanometer. But the native DNA is denatured. There is an increase in the absorbance of UV light. This phenomenon is known as hyperchromicity or hyperchromic effect. The relative absorbance of double stranded DNA at 260 nanometer is 1. The relative absorbance of single-stranded DNA at 260 nanometer is 1.37. The next effect of denaturation of DNA is TM of DNA. The temperature at which the DNA is half denatured is called as melting temperature or TM of DNA. Melting temperature can be monitored by measuring the absorbance of UV light at 260 nanometer because single stranded DNA has higher relative absorbance at this wavelength than the double stranded DNA. DNAs with high concentration of guanine and cytosine are more stable and they require higher temperature for denaturation because three hydrogen bonds are present between guanine and cytosine. Therefore, the TM is also high. DNAs with high concentration of adenine and thymine denatures at lower temperature because only two hydrogen bonds are present between adenine and thymine. Therefore, TM is low. Decrease in viscosity. It is also one of the effects of denaturation of DNA. When the double-stranded DNA is denatured, there is a decrease in the viscosity because the hydrogen bonds between the base pairs are cleaved. Before denaturation, there is a higher viscosity due to the presence of hydrogen bond. Let's see about renaturation or annealing of DNA. The denatured single-stranded DNA can be converted to double-stranded DNA when it is cooled. This process is known as renaturation or annealing. On cooling, renaturation of DNA occurs. Annealing or renaturation occurs only at a temperature below Tm of DNA. So here we can see in the diagram double-stranded DNA is there. When it is subjected to heat, that is denaturation, the double-stranded DNAs are converted into two single-stranded DNA through the break of hydrogen bonds that are present between the two strands. When the single-stranded DNAs are subjected to cooling, then again the hydrogen bonds are formed between the two strands, that is in the base pairs, and the single-stranded DNAs are converted into double-stranded DNA. This process is known as annealing. The annealing or renaturation occurs in two stages. The first one is nucleation and the second one is zippering reaction. So let's see about nucleation reaction. This is the first stage which is slow since the two strands must find each other and form hydrogen bond between them. The next one is the zippering reaction. This stage is fast in which hydrogen bonds are formed between the remaining bases.